Oh, right. Actually, I forgot a couple of things that I needed to point out. Before we move on. Let's get rid of those. So the only other thing that needs pointing out is that these distances are worked out on the assumption that there's going to be an equal rate of recombination between any given, point on the chromosome. And that's not actually the case, right? And so recombination occurs normally on the arms, right? So the kind of the bit where most of the genes are, but the center of the chromosome and actually all the very, very ends as well does not undergo much recombination. There aren't very many genes in there because it's almost always shut down as heterochromatin. But also there's very little recombination that occurs around the centromere or the central area of the chromosome. And so for the arms, linkage map equals the physical map, like the actual physical distance in base pairs between genes. That applies, right? So that is like a pretty much a one-to-one relationship between linkage and the physical distance, right? Or between the genetic map and the physical map. That breaks down in the center of the chromosome, right? So that's just something to kind of keep in mind. It's not a major deal because usually it doesn't really have any effect. Most of the genes we care about are on the arms, but it is worth just kind of keeping in mind. Okay, so now we kind of change gears completely. Now we start getting into a bit of the molecular biology of DNA replication and the like. And so we're going to go through essentially basic recap on the structure of DNA, which should be pretty straightforward to all of you. Then we're going to look at the different modes of replication, like in a kind of general sense. We look at prokaryotic versus eukaryotic DNA replication, which are kind of similar, but there are some important differences between them. And then we'll look at the kind of the downsides of having linear chromosomes, right, which is what eukaryotes have. And I think we look at the actual mechanism of homologous recombination, and I think it's time for, yeah, so then it's time for exam 2. So basically what we're talking about today, on Thursday, and any catch-up time on Tuesday as necessary, because we do start the next chunk on next week on Tuesday, just because that's the way the schedule worked out. Okay, so the next exam opens this time-ish next week on July 6th. So you get to have a lovely July 4th studying genetics. Aren't you happy about that? That looks like that's a yes, right, Robert? You got your head tilted sideways? Okay, so let's start working through it. So what I hope to do today is get through this one at least, like the basic stuff, and then we can get into some of the details on Thursday and start homologous recombination. We'll see how far we get with that. Okay, so again, just a little bit of kind of nomenclature, kind of names of things. Again, we have our bases called the nitrogenous bases. 2 different groups, purines, which have a double ring, and pyrimidines with a Y in, hence thymine and cytosine, they have the single ring. And those, what we colloquially call bases or base pairs in general terms, in terms of distance, we're really talking about a nucleotide. right? Nucleotide is a sugar, a base, and a phosphate group. Nucleoside doesn't have the phosphate. I'm not sure where we'd ever find those, but anyway. And again, another really important difference between DNA and RNA, and we'll actually see where this has an impact or where it's used much later when we talk about RNA processing, is this second prime carbon has an OH group in RNA. But the 3' carbon, this one here, is what is used for the extension of DNA and RNA. That one's the critical one. Sure. The 3' OH and 3' equals the number. Oh, forgot the rest of it. Is one used for of DNA or RNA and DNA strands. That's the one where the new nucleotide is added to. The 2' OH is found only in RNA and is used for funky RNA activities, which we'll come to later. Another bit of recap. Again, DNA is a linear polymer, has a direction, 5' to 3'. That direction is designated basically by whether there's a phosphate group at the end, in which case it's 5' because it's added to the 5' carbon, or if there's an OH group at the end, in which case it's added to the 3' carbon, or it's attached to, rather. Right, so we have direction. That direction is super important for DNA replication, for one, right? So new bases get added on down here. And two, that's the direction in which it is read in making mRNA, or just RNA in general. And these different nucleotides are attached together by phosphodiester bonds. Again, that's a word that we've come across before. Okay. Again, as recap, anti-parallel, right? So one end is going, one strand is going in one direction, one strand's going the other one. That's going to be something we'll hit as an issue in DNA replication in either today or on Thursday. And we have hydrogen bonds kind of sticking these two strands together via the bases. And so this base pairing is the, well, A is how the DNA has its structure, and right, and how RNA is made using the template strand. But it's also kind of really, really important in error-free DNA replication. right? So making sure that when we copy our DNA in for new daughter cells or germ cells, the DNA that those cells inherit is a faithful copy of the original. Otherwise, given how many times we divide, we'd accumulate an enormous number of mutations just in the process of us growing, development. Now, another key point, again, to remind you of is that the number of hydrogen bonds between different bases is not the same. So between A's and T's, adenines and thymines, we have 2 hydrogen bonds, and that's because that's the maximum number that can form, right? If you look at the actual atoms and the properties of each base, even though thymine could potentially form 3 hydrogen bonds, right? Because it has 3 kind of, what would that be? Those would all be, well, that would be electronegative, electronegative, and electropositive, right? So there's 3 potential sites of electro difference, I guess. Adenine only has 2. And those line up very nicely so that the electropositive of the hydrogen on the adenine lines up with the electronegative of the oxygen on the thymine and vice versa. Guanines and cytosines now have 3 because they have 3 potential areas that can form hydrogen bonds. Again, don't forget it's like that, right? So again, they're actually kind of, one of them is upside down relative to the other. If they're both the same way up, you know, it wouldn't work. And so this is actually a very important property of DNA, right? Determined by its sequence. You making popcorn, look? Amor. Amor. You making popcorn? You making popcorn? Oh, just wondered what the banging sound was. Thought something was exploded in the kitchen, and it was popcorn. Yeah, sorry, I can't provide popcorn to students. It's just not allowed. Anyway, so this is really important because if you want to open those strands of DNA more readily, so this would be potentially a site of DNA replication or of the initiation of transcription, you'd want more A's and T's, right? So when we look at the different sites where transcription is initiated, or even DNA replication too, but transcription, you'll find they have mostly T's and A's, right? Now on the flip side, say you're a bacterium called Pyrococcus furiosus, which is such a cool name for an organism. They live around 115 degrees Celsius, which is a big number in Fahrenheit. So give me just a second, Adam. And so they have a more GC rich genome, right? Because their DNA is constantly trying to be shaken apart by all that thermal energy. So they need more G's and C's to hold it together because there's more hydrogen bonds. So Adam, that's, it's kind of a subtle difference really. So G's and C's will obviously break apart too, right? Because, you know, they're still only held together by hydrogen bonds. Quick, let me just give me a second, Rose. And so it's kind of it's not a massive difference, but it's a small but important difference. So if you want to more easily break open a bit of DNA, you would expect there to be more A's and T's there because it takes, you know, essentially 2/3 of the energy of it would if it was G and C's, right? So if you bend DNA in a particular way, you can actually pop open the 2 strands. And that's more likely to happen if there's only 2 hydrogen bonds between each base rather than 3. Yeah, but that doesn't stop G's and C's being pulled apart during transcriptional DNA replication at all. Just means you're more likely to find those areas of opening with A's and T's. DNA editing, splicing. So talking about like CRISPR-Cas9 stuff, it doesn't make any difference, because that you're actually breaking the backbone. Typically a double-stranded break, depending on how you do it, but you're actually breaking the phosphodiester bonds. You're not really focusing on the hydrogen bonds. Just having a shower with Sophie, love. I don't know. It's chaos in this house, absolute chaos. Yeah, good question though. And also because it's enzymatic as well. So, you know, what we're talking about here is more just a basic physical or physiochemical property. If you have enzymes involved in it, just doesn't matter as much. Okay, again, hang on, have I got this? Nope. Right-handed double helix. We've got a major and minor groove, right? So the major groove, the twist is a little bit more open. Minor groove, it's a little bit more closed. This is actually a property that's recognized by proteins that bind DNA, not just the sequence of DNA, but also the actual shape of the DNA too. And then a little bit of terminology. So that's for kind of, get off of you. Don't need to see that. Here she comes. Oh, that popcorn does smell good actually though. Not really a huge popcorn fan unless it's like caramel corn. Love that a lot, but it smells good though. So now we've kind of basically in a bit of a whistle stop tour covered all the basics, right? So these are things which, you know, we already know essentially, and it's yeah, I agree, Adam. But it's important to kind of remember that stuff because we're going to be using a lot of those terms. We're going to come to them a bunch of times. But now another little bit of terminology. Now we're going to be talking about DNA replication. So we really care about where it starts and what it looks like once it starts. And so the replication origin, or which is often called the origin of replication, is a specific site where DNA replication begins, right? In bacteria and viruses, there's typically one of these. In organisms with linear chromosomes like us, there may be thousands. And these aren't random sites. They are sites that have a particular sequence. They're recognized by different proteins, and that's where replication will begin. They are also, again, typically AT rich because it takes a little bit less energy to open up the double helix at those points. Once that whole replication machinery gets going, then we have a replication fork, or typically two replication forks, depending on what organism we're looking at, or more actually. But for each replication origin, you either have one or two replication forks, depending on whether you're, well, unidirectional or bidirectional replication. And that's where all the magic happens. And then just to add to that, actually, I should probably just meant to do this last time. So that replication bubble basically consists of one or two replication forks, expands as DNA replication proceeds. So that's just another kind of term that I use, which I think will be worth remembering. And I'll upload a new version of this to Blackboard when I'm done. Okay. Other point that's worth pointing out, or some points, is that DNA replication is semi-conservative. So each strand of the double helix serves as a template. And you can see that here. So we have the original double strand. This point here, where they're split apart, that's our replication fork, right? Replication origin is presumably kind of down the page some way, right? Don't actually know how far. So here's the replication fork. You can see that these two strands are split and separated, right? They're kind of literally pushed apart. And each one is having newly DNA synthesized, made in that complementary fashion. So when DNA replication is finished, you know, however long that takes, you'll end up with two identical copies. These are going to be your sister chromatids, if this is a eukaryotic organism. And each sister chromatid will consist of one template or parental strand and one daughter strand. So one old and one new. And this replication fork goes along the original DNA double helix, and both of these daughter strands are synthesized at the same time. Right, the synthesis is actually tied together, like the enzymes that do both strands are physically located relative to each other. But DNA, the new nucleotides, have to get added to the 3' end. So if you look at the strand on the left, right, the next one to be added will be another G, and then a T, and then a T, right? So the replication is going up. The next base, we don't actually have the matching DNA here. The next base on the other strand is going to be attached to the 3' end of this adenine. Can anybody see what problem we now have? Right, replication of both strands proceeds at the same time in one direction, but because of the anti-parallel nature, replication actually physically exists in opposite directions. So somehow we have to square that. So even though this strand is getting synthesized in the opposite direction of the movement of the replication fork, we need to make sure it's synthesized at the same time and the same rate as the other strand. So what was the question, Adam? So once the strands have been, like the template strands have been copied, is synthesis finished? No, once these are a double helix, they're a double helix. That is now the new chromosome, and this is now another, well, chromatid, technically. So the original strands never go back together after DNA replication. Each copy now consists of an original and a new strand. Yes, there can be double forks. It just depends on the organism, but we'll get to that later. sometime today, would be my guess. So in DNA replication, obviously there's a whole bunch of things that need to happen. So we obviously have to begin the process, right? And that's going to be signaled by essentially other proteins that control the cell cycle, right? That are going to control when this occurs. We've got to unwind the DNA, right? So that it's actually physically available for the replication machinery. There's going to be elongation. And then termination consists of obviously the physical end to DNA replication, but there's also a whole bunch of other stuff that has to go on afterwards, which we'll get to in a little bit, kind of basically tidying up the mess left behind by DNA replication. And for DNA replication to actually occur, we need a bunch of different things, right? And so if you remember back to the cell cycle, most of these things are actually created during the gap1 phase, right? That's why it's called the gap phase. It takes a long time to make all this stuff. Right, so we need a whole load of DNTPs, and so N stands for any, basically. So that's going to be the adenine, cytosine, guanine, and thymine. We need a bunch of energy, a bunch of ATP as well. And we need a whole load of enzymes, some of which are listed here. And we'll go through what these different things do when we get to that point. And also we need a template, right? And that template has to be single-stranded because we're going to be synthesizing the second opposite strand during DNA replication. So that's why we need to split the DNA helix and separate those two strands. And so all of these things, the DNTPs and the enzymes and the like, those are synthesized during gap1. Obviously, the S phase DNA replication does take time too, but all the preparations for DNA replication occurs in G1. Okay, so now we get to the different ways in which DNA can be replicated depending on the type of organism or nucleic acid material even, right? Because I'm a little hesitant to call a virus an organism, kind of one of those gray areas, right? And so essentially it depends on what type of chromosome you have, whether it's linear or circular. And technically some viruses have linear chromosomes too, but we tend to just not worry about those too much. And we have two different central types. We either have circular DNA, where the DNA exists as a literally a loop circle, even might be a very, very large one, but it is still circular. Start one bit and keep going. Eventually you'll come back to that same spot. And linear DNA, and that's really the premise or the domain of the eukaryotes. Oh, my third dog, who's just had her breakfast and a shower. It's like doggy hotel. And for circular DNA, you can copy it in one of two ways. Either what's called theta replication, which can be either uni or bidirectional. That's kind of getting back to, I think, Adam's original question about if we can have two replication forks. The answer is yes, but again, it depends on which bacteria we're talking about. And there's also rolling circle replication, which is really more for viruses and also plasmids, which are like a form of extra chromosomal DNA for bacteria. So what they carry there, well, many of them carry like virulence determinants on, you know, antibiotic resistance genes, toxin genes, stuff like that. Oh, now that I have it, let's see if I can. Yeah. Good thing about little dogs is that they're very easy to pick up. Although, again, with big dogs, you don't need to pick them up because they can jump on stuff themselves. She's a very needy pup. So every dog has to sleep with someone, usually a child, because otherwise they just howl and howl and howl, and then we don't get any sleep and we're super cranky the following day. So in bacteria, this is most bacteria use theta replication. I'm not sure of any which don't, but I'm also not a microbiologist either, or at least a bacteriologist. And it really depends on which bacteria we're talking about, whether we're going to have bidirectional or unidirectional replication. And so unidirectional is really, there's just one replication fork. For bacteria or circular DNA, there's always only one origin of replication. That's funny, Rose. It's going to be funny actually see what our new pup grows up like, because we were told that Luna, the brown dog that I just showed you, should be like a 40-pound dog, like 70 pounds. So we'll see what happens with the other one. Not much you can do about it anyway, other than just not feed them, which is obviously wrong. So theta replication is called as such because it actually looks like the Greek letter for theta, right, which is kind of like a circle with a line across. And so if it's unidirectional, then that replication fork just goes all the way around and it ends up getting back to the start, the origin of replication. If it's bidirectional, then you have two forks and those spread. Okay. Oh, for sure. Yes, all of this stuff will be uploaded. It's been a bit of a hard class today. So yeah, and if you have any questions, let me know. Hey, get out of it. Sorry, not talking to you. Talking to this dog. Okay, and so obviously with bidirectional replication, now those two replication forks kind of spread further apart until they end up meeting pretty much on the other side of the circular chromosome from the replication origin. And this here, what you see with the two replication forks, now that's a replication bubble. Right, and that just kind of expands and expands until eventually you have the two new copies of the DNA. You are such a needy dog. Okay, so for viruses things as well, it's worth kind of keeping in mind the actual biology of what's going on, right? Viruses replicate fast, really fast. Usually it's a race between the virus and either the kind of cellular immunity of the cell that it's infecting, or the actual immunity of the organism it's infecting, right? And so viruses want to replicate really, really fast, and they don't care quite as much about replication fidelity, right? Hence, the rise of mutations in viruses, particularly RNA viruses, like COVID, for example, Corona or HIV. So instead, what viruses and or some viruses, not all of them, but some of them, do is we have this thing called rolling circle replication. And so essentially what happens here is you synthesize one strand, not the two strands, just one, right? So you make a nick and you cut the backbone of one of the strands, and essentially you kind of peel it off as you synthesize the, yes, yes, yes. Sorry. This thing is being very needy. Um, you peel it off while you're synthesizing that one strand, right? So this is you don't see this at all in eukaryotes, and you don't see it in uh, the normal replication of prokaryotic DNA, right? The main chromosome, I guess one could call it. And so once you have that, now you have a replicated, well, one strand at least, right? So you now have a uh, copy, right? And you have the linear bit of DNA which was peeled off, right? That was replaced by the newly synthesized strand that you see here in red. That linear piece gets circularized, so it gets stuck back together, and now that serves as the template for the other strand, right? Essentially, the inner strand that you see here then gets synthesized later. So essentially what you're doing here is you're synthesizing one strand at a time. Actually, each replication circle can produce multiple strands, Adam. So this can keep on going over and over and over and over and over again. And you can have tens or hundreds of these linear bits because you just keep going around and repeating the same process. These linear bits then have to all get circularized, but then as soon as they do, they can serve as the template for the other strand. So the one at the bottom, Adam, is the original replicate. So if you see here, so if this cycle is not repeated, right? So you just peel off the outside strand essentially and copy that and then stick it, seal it back together. You'll have the newly synthesized strand on the outside and the fresh and the original one on the inside. Now, if that isn't used again, this is what you'll end up with. And then you'll have, one other outer strand to get circularized to be turned into this. It just depends on how many rounds you go through, right? And essentially you can go through multiple rounds to generate these linear bits. And that's really what it's all about. It's about speed. Because while you're synthesizing this outer strand, right, you generate one of these linear strands that gets copied. That can be going on while you're again doing the same thing again, and you can just kind of do a lot of DNA replication in parallel. So that's the big deal about rolling circle replication is speed. And also because, in viruses you want, and also the same for plasmids too, you want multiple copies, right? in prokaryotes, or even in eukaryotes, you want one copy, right? So you end up with two copies. You want to copy your chromosome once, right? So you end up with two copies. Oh, it's not Snowball, sweetheart. She's on my lap. Sorry, puppy's going. I think our puppy found one of our cats, which sounds like fun. Whereas viruses, obviously, if you're a virus, you want to make as many copies of yourself as you can before you either lyse the cell that you're in, or you get found out and the cell you're in gets killed, right? And so the aim here is to produce multiple copies of the genome, and rolling circle replication allows you to do that. Does that make sense? Kind of have to take into account the context of what's going for it to make sense. Now, in eukaryotes, it's totally different. Because in eukaryotes, we have linear chromosomes, right? So bidirectional replication is a given. But what is a kind of an extra wrinkle, right? Those chromosomes are huge, absolutely massive, right? Hundreds of millions of base pairs long. Now, if you just had one origin of replication, yeah, I know. If you only had one, that would take forever to replicate that whole chromosome. It takes long enough as it is anyway, right? S phase is a big chunk of interphase because it takes time to do all this and do it properly. And so as a result, eukaryotic linear chromosomes have multiple origins of replication. Did Pepper find one of the cats? Oh, barking at you. Found my daughter. There you go. And so you can potentially have thousands of these sites of replication in eukaryotic linear chromosomes. And essentially those form many bubbles which merge when they hit each other. So if you look at this, essentially we have one replication origin here, we have another one over here, right? So you have your kind of bidirectional replication. Those forks expand outwards as that DNA is synthesized. The same occurs from the other origin of replication. Forks expand outwards, and when those forks hit each other, essentially, you then have one large replication bubble. And this happens all the way along. So that large replication bubble is going to start expanding. It's going to hit another large one. And it just keeps happening until you end up with two distinct copies, your sister chromatids. And so that's how eukaryotes have solved the issue of size and time, right? Rather than having just one. And in prokaryotes, their chromosomes can be reasonably sized, but I think maybe 1 million bases at the very most. Probably not even that, like in the hundreds of thousands is generally a fairly large bacterial chromosome. And so you can get away with just having one origin of replication. But you can't get away with that in eukaryotes. You need to have multiples to be able to finish everything in time. And again, you can kind of contrast E. coli, when it's in its exponential phase of replication, you can divide once every 20 minutes, give or take. Humans, our cells divide at best once every day, 24 hours. Just goes to show how much more stuff has to be done for that cell division to occur. Anybody got any questions so far? Okay. So again, just to reiterate, new nucleoside gets added to the 3' end. That phosphodiester bond gets formed using the energy in the diphosphate that gets released. But because this is at the 3' end only, and DNA is anti-parallel, essentially we're going to end up with 2 replication bits, yes, in a second, going opposite directions, even though the whole replication fork has to go in one direction. And we basically solve that by having what's called a leading and a lagging strand. And so we have, again, this replication fork is going in one direction. The leading strand is synthesized in the same direction as the replication fork movement. And that's because it's being synthesized from 5' to 3' in that direction. And so you have the replication origin, you start synthesizing the leading strand, and that leading strand will be synthesized continuously in one long go until you hit the next replication fork coming the other way. So that could be in the tens or hundreds of thousands of base pairs long, that leading strand. Now the other strand, so sorry, Snowball, gonna have to put you down. Yeah, she's like, what? I was comfy there. Yeah, I'll bring you back up in a second. So essentially the problem is this. Right, you have 2 strands going in different directions. So if the replication fork was going that away, that's supposed to replication fork. Then the new strand going in that direction can be synthesized continuously because new nucleotides always have to get added to the 3' end of the new DNA strand. So all DNA is synthesized 5' to 3'. That's a given. But the problem is how that ties in with the direction of the replication fork. Ah, dang it. And so this strand, so this is the leading strand. You know, it's funny, I once called T-Mobile or Sprint or whatever you want to call it. And I don't know where the customer service lady was based, but I could hear chickens crow in the background, which I thought was kind of neat. So The other one has to be synthesized that way, right? Because it too has to be synthesized 5' to 3'. But because this is 5' to 3', this one has to go in the opposite direction. So for the lagging strand, you synthesize a little bit, and then you stop, and you kick the DNA out, and then you synthesize another little bit. I just had to turf her off the couch, love, because I need to use my whiteboard. And then you kick it out, and then you synthesize another little bit, right? These are called Okazaki fragments. And in general, this is called the lagging strand. Now, one common misconception is that the lagging strand takes longer for DNA replication to occur. Now, that's not technically true. DNA replication occurs at the same rate for both leading and lagging strands, right? Because it has to. Those two clumps of DNA polymerases are tied together at the replication fork. Now, the issue is we now have lots of these gaps to fix and fill and stick together. That's what takes time. That's why this is called the lagging strand, because it takes longer for this strand to be completely finalized, so it's all done. The actual DNA replication part does not take any different amount of time. It's all the other stuff afterwards. All right, our loner dog has shut up, which is good. It's weird because our other dogs don't bark, and certainly Luna doesn't bark. And so having a dog that barks and stuff is like, oh, that's kind of funky. And so now when you look at it from this single origin of replication in the middle, let's get rid of that to make it a little bit bigger. Right. Now we have our bidirectional replication. Each one of those replication forks has its own leading and lagging strand. Leading strand that's synthesized in the direction of that replication fork movement. So you can see on the going left, it's the top one. Going right, it's the bottom one, because obviously the original template is anti-parallel double helix. And then also each fork has its own lagging strand too. That's the one that's synthesized essentially in the opposite direction to the direction of the replication fork. And again, you have the same deal in bidirectional theta replication, right? Again, we have a lagging strand and a leading strand and a lagging strand. However, you do not have that in rolling circle replication. There is no lagging strand. There's a single origin replication, and you essentially only have a leading strand until that leading strand goes around back to the origin, and this strand being kicked off, one of the original strands, gets circularized, and then you have again a single leading strand synthesized using that as a template. So there isn't the issue of the lagging strand with rolling circle replication, because only one strand is synthesized at a time. Oh, and one thing to point out too is these Okazaki fragments vary in size too. So in eukaryotes, even though the eukaryote chromosomes are larger, the Okazaki fragments are much smaller. I don't actually know why, to be honest. That's a mystery. Ah, come on, you turd. There we go. All right. Does anybody have any questions about like the general basics?
So in other words, the lagging strand is always going to be facing the opposite direction of the leading.
Correct. Because it has to
And
be.
it'll be the same if the direction of movement for replication was the other way around.
Yeah, it's purely determined by which strand is going 5 to 3 prime in that direction and which strand is going 5 to 3 prime in that direction. So if you split that apart, the leading strand is always going to be synthesized 5 prime to 3 prime. So whichever way round you have it, right, doesn't really matter. Oh, someone's at the door. Whichever way around you have it, the strand which is synthesized 5 prime to 3 prime in the direction of movement of the replication fork, that will be the leading strand. And so it's a little bit of an oversimplification to say that this is synthesized backwards. It's not. It's the DNA, the template strand is actually flipped around. So it is synthesized in the same direction. It's just done so backwards. Kind of a little bit complicated to explain. But you don't need to worry too much about that. You just need to know that the lagging strand is synthesized in fragments, small ones. Ah, Mr. Robert is back. Did that make sense, Adam?
Yes.
Cool. Okay, so that's that for this PowerPoint. Yeah, let's save that, whatever I did to it. Now we're going to get into some of the details. Right, so essentially this is kind of going back, but now we're going to look at the actual mechanism of what happens. And so what we're going to do is look at prokaryotic DNA replication first, because in many ways it's kind of a little simpler. So we can get the general principles of what's going on, and then we'll look at eukaryotic DNA replication. And so there's a few extra wrinkles there, not many. And then there's a major one, which is what's called the end replication problem. And this is actually quite a nice image because it really has like everything in there. You know, it has the different, this is actually for eukaryotic replication. We've got our DNA polymerase, we've got primase, we've got RNA primer, we've got our helicase, got all kinds of stuff going on. And so this is actually what makes this particular topic a little bit harder, right? So with, you know, 3-point crosses and the like, you just have to learn the rules and work through it and you get the right answer. When we start talking about molecular biology, now we start talking about a lot of words, right? A lot of different names for things. And so it is difficult to remember all of this stuff because it can literally be an alphabet soup. And so what I always recommend is try and remember, what do these things do? Like, why are they important? What challenges do they help the cell overcome in doing their job? Right? Think about the context. That always helps. Makes a lot more sense when you put these things into context. And that goes for any of the subject, to be honest. polymerase are the ones that add the new nucleotides to the growing strand. So the polymerases are what synthesize the DNA. And so the trick with enzyme names, it's what they do plus ASE or A's. So DNA polymerase makes the DNA polymer. Helicase is all about the helix. Ligase is all about ligating things together. So that's kind of like a grammatical shortcut, I guess. So the first thing that has to happen is we need to create space where these enzymes can get in. We need to create single-stranded DNA, right? That's the kind of first requirement. And in E. coli, it's obviously a little different in prokaryotes. But in E. coli, we have this origin of replication, and just downstream of this origin of replication, we have an AT-rich stretch of DNA. And so essentially what happens is these initiator proteins recognize this origin of replication, this stretch of DNA. And what they do is they actually coil it. They force it to kind of roll up. I don't know who that is. They'll have to wait. Make sure I don't forget it underneath the couch cushion, which has happened before. And in rolling up and bending that DNA, they actually force the AT-rich sequence to actually pop open. But it's kind of like if you imagine if you take a piece of rope and you twist it the other way to the helix of the threads, it forces those threads open. And that's essentially what's going on here. So by forcing the DNA to wrap around those initiator proteins, now you pop open the DNA and now you have the single-stranded DNA which you need. So one thing is that once that DNA pops open, it's going to want to go back together again, right? It's literally like a series of, like fridge magnets. Kind of, they're just going to want to pop itself back together, or magnetic zipper, which I'm not sure if that's a real thing, but it sounds like a cool idea. And so now we have other proteins, single-stranded binding proteins. These obviously recognize single-stranded DNA, hence the name, and they will bind to that DNA and they will hold it open. So they're like the wedge holding the door open. They keep that DNA single-stranded so that all the other stuff can assemble and start doing its job, right? Once DNA starts getting replicated, then it does. Those proteins are unimportant. They don't matter anymore. But they have to be there at the beginning to hold stuff open. And then the final protein of this initiation stage is a protein called helicase. Right? And helicase is the unwinder. So it actually forces the double strands apart to create single-stranded DNA. Once it's able to access, that point when the DNA is opened up and held open by the single-stranded binder proteins. So once you have these conditions down here, single-stranded DNA held open, helicase present, now we can start the process of DNA replication. However, the only wrinkle here is, has anybody actually ever done this? I don't know if you ever wanted to unwind a rope, but if you stick a pencil or something similar or a nail in a rope and you hold one end, like with your foot, I guess, and you try and open up that rope, like to separate into its strands, what happens? What happens if you try and do that? That's pretty funny. I appreciate the knowing about that. Sorry, just got a funny message from someone else who's also having pet issues. Good to know I'm not the only one. So what happens when you try and uncoil a rope? What's going to happen to the downstream? Oh, that's the cat. That looks like the cat from oh, what had Dr. Evil? Austin Powers. He looks like an evil dictator's cat. Maybe. That kind of comes off the wrong way. Sorry, Robert. I wasn't calling you an evil dictator, but it's kind of like if I were an evil dictator, I'd want to have a cat that looked like that, you know, to fit the stereotype. Anyway, so if you, Austin, you know what's funny, Adam? My parents grew up in London, so I've been exposed to what's called Cockney rhyming slang, and I can actually understand some of what he says in Cockney rhyming slang, which is funny. Yeah. Just give you a quick example because it's funny. So that trouble and strife is the Cockney rhyming slang for wife. That always makes me laugh. Oh, another one. This is because you're all American, I'm guessing at least. Septic tank equals yank because septic tanks are full of. So yeah, if you ever go over to England and you're in London in the East End and someone calls you a septic, it's because you're American. So your septic tank. Anyway, you don't need to worry about that. That's not going to be on the exam. So what happens when you try and uncoil something that's coiled, be it rope, be it DNA, whatever, is by uncoiling it in one part, you coil it more in another part. And so actually you could just go and try this. Find yourself a bit of rope that's, you know, not being used to hold something important up and try and unwind it by like forcing a nail or a pencil down it. What you'll actually find is as you unwind it, the free end will spin round and round and round and round and round like that. If that end isn't free, i.e. it's part of a bacterial chromosome, what will happen is that the DNA downstream of that being bit unwound will wound tighter and tighter and tighter. It's actually called supercoiling. And so as that helicase kind of goes out and forces that DNA apart into single-stranded DNA to allow the DNA replication machinery access, the DNA outside of the replication bubble is going to get wound more and more until it just can't be separated. And so both prokaryotes and eukaryotes fix that by something called topoisomerases. And they basically relieve the supercoiling. And so in bacteria, they're often called gyrases. It's the same thing. It's part of the same family of enzymes. And so what DNA gyrases do, or topoisomerases, and there are different ways you can do this. Essentially, you have your double-stranded helix like so, right? And this is replication forks over here, double strand. And so what DNA gyrase does, it can either break one strand, and then there's a whole bunch of energy kind of coiled up, right, in that supercoiled area and ahead of the replication fork. As soon as you break it, it spins round. And then the gyrase sticks it back together again. And so by doing so, it relieves that coiling, that supercoiling ahead of the replication fork. Now, some also do a double-stranded break, in which case it's a lot simpler to visualize. If you break the DNA, now you have a free end. right? And that free end is now going to want to spin, and you just stick it back together again. Once it is, that energy has been released. And so this, on both replication forks, you have to have a DNA gyrase or topoisomerase, pardon me, moving ahead to release that supercoiling. Otherwise, you just get like a scrunched up, tangled mess. So DNA gyrase equals topoisomerase. Basically, it cuts the DNA ahead of the replication fork to release the supercoiling induced by the helicase, separating the two strands. So again, every, it's kind of like parroting Newtonian physics. Every action has an equal and opposite reaction. So if you're opening DNA, essentially you're trying to close DNA more elsewhere, right? Because you can't close DNA any more than the double-stranded helix, essentially you're making that DNA twist tighter. And so the more you open the DNA, the tighter the DNA ahead of it is going to twist. And so this topoisomerase or DNA gyrase, they're essentially one and the same thing, gets rid of that by cutting the DNA and allowing that DNA to rotate, releasing that coiling, and then they stick it back together again. Which is pretty cool. Okay, I think that's enough for now. We're pretty much bang at the end. Read ahead between now and Thursday. You'll get some terminology and stuff kind of into your head. And we'll continue the mechanics of this on Thursday. And then go over homologous recombination. And now my stomach is telling me it is time for lunch. So I'm going to take a break, as should you all. I'm going to go and rummage through the pantry and see what I can find that might be interesting to eat. And I'll see you back in half an hour, 2 p.m. And we'll go over anything needed for today's deadline for the Excel spreadsheet and also the mini lab report in general. Cool. All right. I hope your cat behaves itself, Robert. And I shall see you all later.